Right, so for today, our topic is actually the chemical foundation of life. Talking a bit about chemistry, we got to start at the beginning here, which is the idea that atoms are the building blocks of molecules. And again, the reason why we are talking about chemistry in a biology class is that biology is in some ways built upon chemistry because life is actually composed of matter, right? Matter is everything around us that occupies space and has mass. Now, if we break down everything kind of to the smallest building block of things that we typically encounter in the world around us, we have atoms, and atoms exist as different elements. So elements are unique forms of matter that cannot be broken down into other substances that have specific chemical or specific physical properties. I like to think of elements as a sort of type of atom. So again, these elements cannot be broken down into smaller substances, and each one has a name and is designated by a specific chemical symbol that is 1 to 2 letters. So for example, sulfur, the shorthand way that we write that is with the letter S. Calcium, the shorthand way we write that are with the letters C and A. Now, there's lots, and we'll get to a picture of the periodic table, which shows all the known elements. But for the purposes of our class here, there are a few that are actually the most important. The four most common elements that are found in living organisms are, first of all, carbon, whose symbol is C, oxygen, whose symbol is O, hydrogen, whose symbol is H, and nitrogen, whose symbol is N. If you look at the chemical composition of living things, and this is particularly human cells here, you notice, right, we're made of 65% oxygen, I believe this is by mass, 18% carbon, 10% hydrogen, and 3% nitrogen, which these elements are very much enriched as compared to the environment around us. There is a significant amount of oxygen in the atmosphere, but less than what we find in cells, right? Same with carbon and hydrogen. Nitrogen, actually, there's a lot of it in the atmosphere and not very much of it in the crust. But most of this nitrogen that's found in the atmosphere is actually in a form that living things cannot use. So, it is sort of a special kind of nitrogen, if you will, that gets into living things because of the action of some microorganisms, actually. So again, returning to this idea that atoms are the building blocks of elements. Atoms themselves, I mentioned earlier that this is the smallest kind of unit that we would see sort of on an everyday basis. The atom itself can be broken apart into smaller parts. In particular, it has a center region that's called a nucleus and contains some particles called protons and neutrons. And then there's an outside layer of the atom that contains another type of particle called electrons. So together those, are considered to be the subatomic particles. Now, the differences between these particles, the proton has a positive charge, and the neutron has no charge, and the electron has a negative charge. The mass of a proton is 1, the mass of a neutron is 1, and the mass of an electron is practically 0. And it's the protons and neutrons that are found in the middle in the nucleus, and the electrons are found outside in what we call the orbitals. So here's a picture, a diagram of an atom where maybe we can see some of these things more clearly. In the middle, in the nucleus here, we have the protons with the positive charge, the neutrons with no charge, and outside here, in this picture, appears to be kind of orbiting around the nucleus like planets orbit around the sun, are the electrons with a negative charge. Notice that in this picture, right, as well as pictures that we could draw of other atoms, to start out with, at least, the number of positive protons in the middle and the number of negative electrons on the outside have to be equal to balance out the charge. Otherwise, this would turn into what is known as an ion, right? So to start out with, the number of protons in the middle and the number of electrons on the outside are equal in an atom. There's a couple of terms here that apply to atoms. that we're going to discuss here. The first one is the atomic number. So the atomic number is the number of protons that are in the nucleus of an atom. And it's actually this, the atomic number, that sets apart different atoms and determines what element they are. So in this previous picture here, we were looking at helium. All helium atoms have 2 protons in the middle, and that's what makes it helium. Hydrogen is a different element. Hydrogen atoms have 1 proton in the nucleus, and that's what makes it hydrogen. The atomic mass of an atom is, if you recall, when we looked at the subatomic particles, we said that protons have a mass of 1 and neutrons have a mass of 1. So the atomic mass is how much the atom weighs, and that's roughly equal then to the number of protons and neutrons. Now, unlike the atomic number, where it's a defining characteristic of that element, all helium has 2 protons in the middle, different helium atoms might actually have a slightly different atomic mass because the number of neutrons that are in the atom can vary. So one thing that we might ask you to do then is to calculate atomic mass based on atomic number. Here's an example. We've got this particular element, carbon, and up in the corner here, that's the atomic number. If you remember, the atomic number is the number of protons. The number at the bottom here, this is the mass number. The mass number at the bottom corresponds to, again, the number of both protons and neutrons put together. And you can see there's 2 different versions of carbon, 2 different kinds of carbon atoms here that exist within this element that are shown here. One that has a mass number of 12 and one that has a mass number of 13. So both of them have 6 protons, but this one, remember this number here is the number of protons plus neutrons. So that means it has to be, to get 12, it has to be 6 plus 6. There must be 6 neutrons in this atom. This one has a mass number of 13. That must mean that there are 7 neutrons to give it that number of 13. Now, what you do see on the periodic table is actually the average mass number of all the carbon atoms. In the case of carbon, that average turns out to 12.011. So, from that, interpreting that number, you can say that probably most carbon atoms look like this. They have a mass number of 12, meaning that they have 6 protons and 6 neutrons. But there are a few that are a little heavier that have extra neutrons, and that's what creates that 0.01. So again, the term now for different atoms that have a different number of neutrons are what we call isotopes. So I mentioned the number of protons is a defining characteristic of that element. So all here hydrogen atoms have 1 proton, but you could have a hydrogen atom that has an extra neutron in it, right? That makes it a little heavier, or you could even have some hydrogen atoms that have 2 neutrons in it that makes it even heavier. These 3 represent different isotopes of the same element. So isotopes can be used as a research tool, particularly in figuring out the age of a sample. So this is what is called radiometric dating, and it takes advantage of the fact that a lot of times some of those atoms that have the extra neutrons in it, some of the heavier isotopes, are actually unstable. So they don't remain the way they are. Over time, they will decay and turn into something else. So in figuring out then, the ratio of these isotopes that are left, you can actually figure out how old a particular sample might be. All right, so I mentioned that we were going to take a look at the periodic table, which again, hopefully you've seen at some point in the past, but here it is to remind you what it looks like. So this is just a way that we can organize all of the different elements that we know that exist, right? So if you look at this periodic table in the top left-hand corner here, that is again the atomic number. That tells you how many protons, and remember in a neutral atom to start out with, then how many electrons each of these elements has. So hydrogen has 1 proton and 1 electron to start out with. Helium has 2. Carbon here has 6 protons and 6 electrons to start out with. This number that exists below the symbol, again, is the average mass number for all the atoms of this particular element. All right, atoms that have a neutral charge have an equal number of protons in the center in the nucleus and electrons on the outside. Now, so far we were just kind of saying, electrons are sort of on the outside. We haven't really looked at how they actually appear in the atom. The model that's actually probably going to be most useful for us here is one that's called the Bohr model, a way of thinking about how these electrons are arranged in these atoms. It's actually an earlier model. There's an updated one, which we'll talk about just a little bit later. And in this, you've got protons and neutrons in the center, and the electrons, again, orbit, go in kind of circles around the nucleus here in different energy levels or different shells. Now, the closest ones to the nucleus have the lowest energy, and the ones that are farther away have greater energy. And the way that electrons fill in these shells is they always start at the lowest energy. So hydrogen, which just has 1 electron, will look like this, right? It'll be the hydrogen atom with 1 electron in the first shell. This first shell can hold 2 electrons. So helium, when we go to that one and look at an atom there, it'll have 2 electrons in this first shell. The next element, which is lithium, is going to have 2 here, and it's going to have its next electron then fill in the next available shell, which here is called the 2n level. So as you get more and more electrons, you get them filling in these shells further and further away from the nucleus. Now here's kind of for a selected number of elements then what their electrons look like according to this model. You've got hydrogen with 1 electron in that first shell, helium with 2 electrons in that first shell. Lithium, now the first shell is filled, so it's going to add an electron to the outside of that. Carbon here, now you've got this shell filled, and it's going to have those 4 electrons then in that, and so on and so forth, right? And you can see the more and more electrons we get, the further and further away, the higher energy the electrons have to take on. Now, what's important for us for the purposes of understanding how atoms interact with each other is this outermost shell. And depending on the different element, right, that's going to be a different energy level. For the smaller ones, it's going to be the ones closest to the nucleus, right? But if you've got more electrons, then you're going to start filling these upper levels. Whichever one is the last one to fill and contain electrons is what is called the valence shell. A general rule of how atoms interact with each other is, and they're not really trying to do things in the sense that you and I do, but you could kind of think about it a bit that way. Atoms are trying to obtain the most stable configuration of their electrons. They all want their electrons in the valence shell to look a certain way, namely that they want their valence shell to be completely full of electrons. Now, the elements in group 18, which are called the noble gases on the periodic table, are the ones that are the most stable because they naturally have a full valence shell of electrons. In general, other than that first layer that we talked about with hydrogen and helium, most of the shells can accommodate 8 electrons. So the magic number for chemical bonding most of the time is 8. It's so, important that there's a special name for that called the octet rule. So atoms are all trying to get 8 electrons in their energy shells. Now, the group 1 elements, the first column of the periodic table, have, if we look at their configuration, right, like sodium here is an example of one of those, right? It's got in its valence shell just 1 electron. So it's really not very happy because it could easily be satisfied and happy if it had the second energy level be its valence shell. So it really doesn't actually want that extra electron there. On the other hand, right, the group 17 elements like fluorine and chlorine here, they're just 1 shy of having a full set of electrons. When they interact with other atoms, that is what they are quote unquote trying to do, is to gain an extra electron. In this picture, there is actually a mistake. The diagram of argon here that's shown is the same as chlorine. It should actually look like this, where there is an extra electron here that would be filled out and have 8 electrons total then in this outer shell. All right, so I mentioned that the Bohr model was probably going to be the most useful for us in thinking about how atoms interact with each other in chemicals, but it is technically not the most accurate. And what the current understanding is that rather than electrons being in planet-like orbits, they're not going in circles really around the nucleus. They are actually found in these complex shapes that are called electron orbitals that are not all, spherical or round. They have these sort of shapes to them. And rather than moving in a well-defined circle, the electrons are actually kind of moving at random within these shapes. We know that with a certain, confidence, the electron is somewhere within that shape, but we don't know exactly where it is. So I mentioned that, the shapes are a little bit actually kind of weird, right? They're not all circular. This is what they actually look like. That first energy level that we saw as a nice circle around the nucleus is actually a sphere. And again, instead of the electron going in a nice circle around here, it's just kind of moving at random. The two electrons that can populate this one are somewhere in there. We don't know exactly where, but we're pretty sure they're somewhere in here. Once that is full with 2 electrons, we move on to the next energy level here, which is also a sphere. In this case, we've got 2 more electrons that can fit in there that are kind of moving at random. We don't know where they are. After that, we move on to some other subshells in the next energy level. And these are actually have this kind of figure 8 dumbbell type shape to them. So somewhere in here are the electrons. We don't know exactly where. This video here that shows you a 3D version of these orbitals being built around a particular atom to give you a better idea of what they look like in 3D. This idea of the electrons and where they are in the atom is going to lead to this idea of how they interact with each other. And atoms interact with each other by combining sometimes or breaking apart in what is called a chemical reaction. Chemical reactions occur because there are changes in distribution of the electrons between atoms. How do we depict chemical reactions? Well, we can do it in an equation that looks like this, where we have on the left-hand side what we start out with called our reactants, and on the right-hand side what we end up with called our products. And typically we draw an arrow in between. If it's a solid arrow like this, that means that this is a unidirectional reaction. It goes this way. There are what are called reversible reactions, where maybe sometimes we can get both of these kinds of reactions happening at the same time, going back and forth. But this one, it seems to be proceeding this way, right? We're taking this chemical, turning it into these 2 different chemicals, which is the next point here, right? So this is an irreversible reaction. A reversible reaction, if there's the double arrows, again means that within this particular, say, solution, both of these things are happening. You've got these 2 coming together to form this. You've got this breaking apart to form these. Now, we mentioned that the electrons are kind of redistributing themselves. Well, what does that actually do then to the atoms as a whole? What it does is it tends to form what are called chemical bonds, which are the attractive force that links atoms together to form molecules. So here's an example, the formation of water. To start out with, we've got an oxygen atom and 2 hydrogen atoms. And if you remember, we said that these atoms all, quote unquote, want their valence shell to be full and to have the optimal number of electrons. Well, hydrogen has just 1 here. And if you remember, its valence shell can fit 2 electrons. So it really wants 2 electrons. Oxygen, on the other hand, in its valence shell, and we're only in this picture showing those valence shell electrons, not showing all the ones in the middle, it's got 1, 2, 3, 4, 5, 6, right? So it's kind of short 2, because remember, according to the octet rule, it wants 8. Well, in this case, let's say that oxygen and these 2 hydrogens get together and they decide to work as a group and strike a deal where this oxygen is going to share its electron with this hydrogen. Now this hydrogen has 1, 2, its valence shell is full. This oxygen atom, right, now has gained an extra electron, so it's a little closer to being happy. If it also strikes a deal with another hydrogen atom, this hydrogen atom now has 1, 2, and now the oxygen has 1, 2, 3, 4, 5, 6, 7, 8, a completely filled valence shell. So it is now happy. Everyone is now happy. The thing about it is, though, because they're now sharing this electron, let's say if you imagine that you and a friend decide to share a piece of paper, and you know you need to keep that piece of paper whole. You're not going to tear it in half or anything. You're both going to hold on to one end of it. Now, as long as you're sharing that piece of paper, wherever you go, your friend has to go too. So in the same way, these atoms are now bonded together to form this molecule. So what we saw in the previous slide is that oxygen was sharing one electron each, right, with two hydrogen atoms. But you can have more than one set of electrons shared between two atoms. In fact, the oxygen that's in the air around us is actually O2. It's two oxygen atoms that are sharing two sets of electrons. They are double bonded together. Now, talking about that previous type of bond, we were sharing electrons, and that is called a covalent bond. There is another type of bond that can occur called an ionic bond, where atoms either give up or gain electrons. So here's the formation of sodium chloride, more commonly known as salt. If you remember, again, just looking at the valence shell, sodium is in that first column of the periodic table. It's got one extra electron in its outermost shell. So it really doesn't want that one electron. It wants to have the full set in the previous level. Chlorine, on the other hand, is short one. So they decide to make a deal where sodium gives one electron to chlorine. Now, sodium has a full set of valence electrons in the shell that's next, that's a little bit more to the inside here. And chlorine has a full set of valence electrons here in its outermost shell. However, because if you remember, electrons have a negative charge, and we started out with balanced negative and balanced positive here. If sodium loses this one electron, now that means there's actually more plus in the nucleus than there is minus on the outside. That gives sodium an overall positive charge, while chlorine, since it got an extra electron, it became more negative. Now, the basis for the bonding in this case, why these two are stuck together, because they're not sharing something like in a covalent bond, is that in chemistry, plus and minus charges actually attract each other. So after this exchange of electrons has occurred, the plus and minus particles will stick together. However, they can be, separated by water, say, which is why, when you mix salt in water, it dissolves. Okay, now there is another situation to build on what we talked about here so far. We said covalent bonds are sharing. Ionic bonds is when one atom loses electrons, the other one gains. There is something that's kind of in between in a way, which is called a polar covalent bond. In this case, it is a covalent bond, meaning there's sharing going on, but the polar part tells us that the sharing is unequal. That means the electrons are actually more attracted to one of the nuclei than the other, right? So water is a great example, and the property that determines how strongly the nucleus pulls on electrons is called the electronegativity. So oxygen has a higher electronegativity than hydrogen does, and that means it wants the electrons more. It's going to pull more strongly on the electrons. The electrons within this bond here are actually going to hang out more on this side than they are on this side. That makes the oxygen just slightly negative, and we use that lowercase delta minus to show that the electrons hang out more with this one than they do with this one, right? The one that's slightly minus, the electrons are more here than the one that's slightly positive. By the way, if we contrast this, what happens in water with what happens in molecules that are made up of carbon and hydrogen actually have a really similar electronegativity. So overall in this molecule, the electrons are kind of equally distributed around. Now again, when it's unequal, we call that a polar covalent bond. And if there are polar covalent bonds, we might get what is called a polar molecule, where overall on the whole molecule, the electrons hang out on one side more than the other. If there are no polar bonds, like in this molecule where it's just carbon and hydrogen, then the whole thing becomes nonpolar. It is actually possible to have, though, a polar bond, as in the case of carbon and oxygen. In this case, overall, it kind of equals out. Because, the electrons are going to get pulled towards this oxygen this way and this oxygen towards this way. But because these two are equal, overall, there's kind of no place on the molecule where the electrons are hanging out more than the other. Now, the consequences of molecules being polar versus nonpolar is it determines how these molecules then interact with each other. So we mentioned that carbon and hydrogen have a similar electronegativity. So things that are made of just carbon and hydrogen, or a lot of just carbon and hydrogen, tend to be very nonpolar. That is the case with oil, right? Water, on the other hand, as we saw, is a polar molecule. Now, polar things like to mix with other polar things. Non-polar things like to mix with other non-polar things, and they don't like to get together. So as you can see here, right, oil and water don't mix. If you've ever had oil and vinegar salad dressing, you kind of have to shake it up right before you pour it on your salad. Otherwise, you just get, like the oily layer, say. Now, there are types of weaker bonds, and in fact, depending on how you define it, they may not really be considered bonds at all, not at least like the way that ionic and covalent bonds are. These are strong bonds that hold molecules together and require energy to break. These weaker bonds are actually what we might consider to be intermolecular attractions. Between two different water molecules, the force that holds those two different molecules together is what is called a hydrogen bond, where the slightly positive hydrogen and the slightly negative oxygen, say in water, interact with each other and hold two different water molecules together. There are even interactions called van der Waals interactions that can hold, non-polar things together, where the electrons from one kind of push a little bit on the electrons of another and hold it together. But these are both, again, weaker interactions and not really true bonds. They're very weak bonds. So I'm going to show you guys a demonstration here. And if we were doing this face-to-face, I would actually have two volunteers come up and do it. Because we can't do that, I had some microbe mascots here do it for me. So here I'm going to show you what happens, right, when there's an ionic bond that forms. Remind you this way. So let's say we've got two different individuals here who are representing two different atoms. This atom here has some extra electrons that it doesn't really want. So what it could do is it could give those electrons away to another atom, right? Now, what happens because of that, this atom here, because it lost electrons, it lost a negative charge, it became positive. This atom here, when it gained electrons, became a little negative. So we formed a positive ion and a negative ion, and the fact that plus and minus are attracted to each other means that these two are now bonded together, and that is the basis of the ionic bond. Here we have our two mascots modeling a nonpolar covalent bond, where again now instead of an exchange, they're going to share the electrons. And since they're both holding on to one end of this rope here, as you can see, right, wherever one of them goes, the other one has to go as well. Now we could also have a polar covalent bond, where again, they're sharing, but it's unequal. So let's pretend that they kind of tug of war a little bit, and the E. coli here won. So the electrons appear a little bit closer to it than it does to the HPV here. So that means that this one is going to be slightly negative, while this one is going to be slightly positive, right? But not a true positive and negative, because they both are still sharing these electrons, right? The electrons haven't totally gone away from this one. Now, again, what is the consequence of that polar bond? Well, if we had another similar molecule that came near this one, because this atom now is slightly negative, if it comes across another atom that's slightly positive, these two are going to be attracted to each other. So notice now we've got two separate molecules that are being held together by this weak force of attraction, a hydrogen bond. Water is actually a very important chemical to life. In fact, water makes up 60 to 70% of the human body and of our cells. It is the most critical molecule for life on Earth. Some things about water, first of all, is that it is a polar molecule and it can form hydrogen bonds. And that is going to determine how other things in the cell interact with it. Right, so again, hydrogen bonds are the key to understanding why certain things are the way they are in the cell, chemically speaking. Right, as we looked at in our example, it's polar. The electrons tend to hang out with the oxygen more than they do with the hydrogen. That makes this oxygen negative, slightly, and it makes the hydrogen slightly positive. And all the water molecules are that way, right? So this one is slightly negative. So you're going to get this force of attraction between the hydrogen on one and the oxygen on the other. Now, as you probably know, water has three states. It can exist as a liquid, which is mostly what we are going to see in cells, but it can also exist as a gas or as a solid, also known as ice. Now, hydrogen bonds make water do several different sort of interesting things. The first one is that when it forms a solid, when it forms ice, ice is actually less dense than liquid water. That's why when you have a glass of water and you throw some ice in it, it actually floats on the top. That's actually not true for most substances. Most of the time, the solid is going to be more dense and will sink to the bottom. But this is actually really good for living things, particularly things that live in the water, because there can be that layer then of ice that forms on the top, and they can, maintain their lives below it and swim around. Some other important things about water and that hydrogen bonding. First of all, because the water molecules have strong hydrogen bonding going on, although again, remember by definition, it is a weak bond, but because the water molecules are fairly strongly attracted to each other, it means that, for example, when you heat water up, it actually takes a long time for water to heat up and a long time for it to cool down. Water sort of buffers changes in temperatures. So the reason for this is, heat is actually related to the movement of molecules. Because they're all kind of stuck together, it actually takes more energy to make them move a little faster. Now, there is actually a way that we measure this property, how long it takes or how much energy it takes to heat something up. It's called heat capacity. And actually, because water is so important, we define the typical unit for this, the calorie, as the amount of heat that's required to raise the temperature of 1 gram of water 1 degree Celsius, right? And we define that as a calorie. Although note that, you can have a specific heat capacity of other substances, like alcohols have a specific heat capacity to them, right? It's just that we kind of define the unit based on water because it's so important, right? But again, water actually takes a long time to heat up or to cool down. That's why when you have, say, an island, it actually takes longer for the temperature to change in the ocean and hence in the areas nearby. Similar to that, because again, the water molecules are strongly attracted to each other, water has a high, what is known as heat of vaporization. So it requires a lot of energy to change 1 gram of liquid water into 1 gram of steam, into gas, right? It takes a lot of energy to do that. Because water is so polar, again, it also is an excellent solvent, meaning that things dissolve well in it. So water is a solvent. The stuff that you dissolve in it are solutes. And again, this could include other polar molecules. It could also include ions that are formed through ionic bonding. So water dissolves lots of stuff, but not everything, right? Not the non-polar things. So again, building on this idea then that water dissolves things, most biochemical reactions are going to occur in water since so much of the cell is made of water. And the chemical reactions depend on the collisions of things and therefore on the concentration, because the more of something you have, dissolved in the water, the more likely it is to bump into something else that it might react with. So as a result for that, here and, maybe in labs, sometimes we're going to be making solutions. So it's important to kind of understand what that concentration, the amount of stuff dissolved in the water means. Now, molecular mass is the sum of all the masses of atoms in a molecule. So we were looking at the mass of the atom, right? And remember, those atoms can be put together to make molecules. So we could have a mass then of an entire molecule. Now, another thing is, we've got, besides measuring the mass of them, we could measure the actual number of molecules that we have. And the unit we typically use for that is what is called the mole. Because molecules are so small, in order to get a usable kind of unit here, we have to have a lot of them. So 1 mole is actually equal to 6.02 times 10 to the 23rd, which if you're not too familiar with math, how to do this is you move the decimal point 23 places this way. So this is a really, really, really huge number, right? Now, the reason that this number is important is because it's defined that this number of Daltons of the mass unit that we were using before, it should be equal to 1 gram. So, what that means is that 1 gram of a particular substance, should have that many molecules in it, right? So, for example, right, sodium chloride, the masses, if we look at the periodic table for sodium and chlorine, come out to 22.99 and 35.45. If we add those together, we get 58.44. So 1 mole, or 6.02 times 10 to the 23rd sodium chloride particles, has a mass of 58.44 grams. Now, how does this apply to solutions? So molarity is the number of moles of solute per liter of solution. So again, what this means is we could weigh out, if we can convert then to the mass, we can weigh out the amount of stuff that we want to dissolve in a given amount of solution, and we can make that solution. So to make 1 liter of a 1 molar solution of sodium chloride, if you remember, we said that 1 mole of that should weigh 58.44 grams. What we would do is we would weigh out 58.44 grams. We would add it to some water, mix it up so that it dissolves, because remember, this is an ionic compound, so water should be able to dissolve it. And then you add some more water until the total volume of what you have is 1 liter. So that's how you would make 1 liter of the solution. How do you think you would make a 0.5 molar solution? You'd use half a mole instead of the whole mole. So divide this number by 2, and that would give you half of a mole. And then you would put that in a little bit of water, and then bring the total volume up to 1 liter. So, like around 29-ish grams, 29.22, I think, in 1 liter. And then that would give you a 0.5 molar solution. We talked about how water does dissolve ionic things, and it's because it's got this positive side to it, right, and this negative side to it. So the positive hydrogens are going to be attracted to negative ions, and the slightly negative oxygens are going to be attracted to positive ions, and that's what causes them to break apart when you dissolve them. The water molecules will arrange themselves around the ions like this in what are called spheres of hydration. So another thing about water that's important, because again, the molecules, key idea here, right? Water molecules are really strongly attracted to each other, and that has several different consequences, as well as being attracted to other polar ionic things. So cohesion, water molecules stick well to each other, right? So drops of water actually hold themselves well together. This creates this, what is called surface tension. So, water essentially kind of forms a skin on it, right? And you can float things perhaps that you would not expect in water because of this, right? In fact, some insects, for example, that sort of walk on the surface of water for this reason. Again, because, water molecules stick together well, they also display adhesion, so they can stick to certain kinds of surfaces as well. And this allows water to be drawn up through what is called capillary action, through, say, like a tube. There's an example of what I was mentioning previously with the insects that can kind of walk on the surface of water. All right, now another topic here, again, related to water, has to do with acids and bases, which we measure through a value called pH. So the pH of the solution indicates whether it's an acid or whether it's acidic or basic, or another word for basic is alkaline. So acidity or alkalinity. Now, in normal water, there's a small amount of this reaction going on. You've got water that just by chance happens to break up into H plus and OH minus ions. And it's a very small amount of this. It's 1 times 10 to the minus 7 moles per liter. But you can add certain things to water that tend to increase the amount of H plus. We call those kinds of chemicals acids, or you could add some chemicals that would increase the amount of OH minus, and you call those chemicals alkaline or basic. The measure that we typically use to talk about whether something is acidic or basic is what is known as the pH. So pH is related to the concentration of the H plus ions in the solution. And, because if you look, this is a reversible reaction. As the amount of H plus goes up, the amount of OH minus is going to go down, and vice versa. So really just being able to measure one of these things kind of gives us a snapshot of this idea of what's going on with the H plus and the OH minus. So pH, the way that we do the calculation, it is the base 10 logarithm of the H plus. The log of 1 times 10 to the minus 7 is minus 7, and the negative of that number is 7, right? So basically to do logs, again, you kind of just take, if it's 1 times 10 to the something, you take that exponent, and then the log is kind of undoing the exponent action, right? So you get like the minus 7 here, right? And because it's the negative log, you get just 7. So that means that neutral is going to have a pH of 7, right? Anything that's above 7 is going to be alkaline or basic. And anything that's below that has a lower pH is going to be acidic. So, I'll let you kind of look at what some of the substances that are acidic and basic are. So, again, here, let me throw this on there just so we're clear. So things that are acidic actually have more H plus to them, right? And things that are basic actually have less H plus and more OH minus. Every time you move up a level or down a level in pH, right, it's actually, because it's a log, it's actually 10 times. When we go from a pH of 8 to 7, a solution 7 actually has 10 times the amount of H plus that the solution with a pH of 8 does. And if you go again, to 6, that's another 10 times. And then if you go, again to 5, that's another 10 times.
Now, organisms, a part of maintaining themselves is also to maintain the internal pH within an organism. So for example, our blood actually has an ideal pH. And, various processes that occur in the body could, either make more H plus or decrease that amount of H plus. And sometimes that might not be good, right? So as a result, there's what we call buffers. So buffers are solutions that tend to maintain a certain pH. Now, the way they do this is by some reversible reactions, as you can see here. So if you look here, if we add H plus to the system, what ends up happening is, there's this ion here that's circulating in the blood that can then bond to this and make this compound here, right? Which can then break up into water and CO2. So, if we add something this way, it'll kind of drive the reaction this way. If we, take away some H plus, say, then it'll drive the reaction this way, so that we always kind of maintain an ideal pH, an ideal amount of H plus. So that's within our bodies. We see that buffering system. That actually, that same buffering system, the same chemical, this is called carbonic acid. This is carbon dioxide and water. That same kind of buffering system exists actually within the oceans here and is affected by emissions of carbon dioxide. Because if you look at what happens in this image, when we add CO2 to the system, say we add more carbon dioxide to the air, then some of that gets dissolved into the water and forms more of this acid here. That acid, right, in turn gives off H plus. Now, that H plus can bond with this carbonate ion here, form this bicarbonate ion here. And one thing that's important in the oceans is that this carbonate ion is found in corals. Say, for example, they combine it with calcium, form an ionic compound here of calcium carbonate. And that's actually what corals make their skeletons out of. So, if we add CO2, what we end up doing is sucking away some of the carbonate that's found in the coral skeleton here in order to maintain this buffering system. And that means that weakens the structure of the corals. Emissions of CO2 end up having this impact. Maybe you've heard of this in the context of climate change or global warming. Not just that, but it ends up impacting this aquatic ecosystem as well. Okay, so moving on to the last section here of our introductory chapter to chemistry, last section of chapter 2, we're going to look more specifically now at carbon, the element carbon, and some of the properties of its chemistry and why that's important to life. Okay, so first of all, carbon is an essential element. It is actually the key component of most of the things in the cell other than water. So carbon is unique because it tends to form covalent bonds, and specifically 4 bonds with different atoms. Or perhaps it can double up and do a double bond with something. If you recall, when we looked at the periodic table, right, carbon is in that column where it's got 4 valence electrons, so it tends to form 4 bonds, right? Now, perhaps the simplest kind of carbon compound that we might see are what are called hydrocarbons. So as their name suggests, they are made up of carbon and hydrogen, the simplest one being methane. So, again, these are all covalent bonds between carbon and hydrogen. And these bonds store energy, which we can see is released when we burn these compounds. So a lot of these compounds are fuels that we use for things, including our cars, or to burn for electricity, say. Now, here's a diagram of methane. As you can see, right, we've got carbon and it's got 4 single bonds to 4 different hydrogen atoms here. Now, the basic shape that carbon takes when it forms single bonds is what we call tetrahedral geometry, where it's sort of like a pyramid shape, if you will, right? If you can imagine, there's sort of these triangle faces on each side here. It looks like this. Now, that was the simplest one, methane. We could add more carbon and more hydrogen atoms to make some larger molecules here. In fact, if we have 2 carbons, then we call that, and all single bonds with hydrogen, we call that ethane. Here we see a hydrocarbon that's got a double bond in it. So again, sometimes atoms can share more than 1 pair of electrons and we see double bonds. Carbon can also actually form a ring structure. So here are a few examples of some hydrocarbons with rings in them. Now, note that these rings on the right-hand side here actually have this alternating pattern of double bonds and single bonds. That's actually a special kind of ring that we call an aromatic ring. So the reason, aroma, if you think about that word, it's a scent, right? Compounds that have that kind of structure in them do tend to have like a strong smell to them, which is why they're called aromatic. So the special thing about this alternating single and double bond is that actually what's going on here, instead of the alternating bonds, these extra electrons that are in the double bonds are actually kind of floating amongst all the members here. So that creates some special chemical properties that those rings do have. So there's another term here that applies to a lot of these carbon compounds in particular, which is the term isomer. So, so far when we've been looking at different chemicals, we might use the formula, right? Where we use the symbol as well as the little number to indicate what is in that chemical, what the different atoms are. But sometimes, that formula is actually not unique to a chemical. Sometimes there could be more than one chemical that's got a slightly different structure that actually has the same amounts of atoms and hence the same formula, right? If you see here, structural isomers, our first type, we've got this molecule and this molecule. Despite having the same number of carbons and hydrogens, they are arranged slightly differently, right, in these 2 molecules. So those are in fact 2 different molecules. Next kind of isomer, geometric isomer, applies to when we have a double bond, as I mentioned, because this double bond geometry does create these 2 different possibilities. In this case, you could have the hydrogens on the same side of the double bond and the methyl groups on the same side, or you can have them on opposite sides. We call the same side the cis configuration, and the opposite side's the trans configuration. Last type of isomer is what we call enantiomers. So enantiomers, they share the same formula and they actually look pretty similar, but these 2 are actually mirror images of each other. And this occurs if you have a carbon with 4 different things bonded to it. You could have one of 2 different kinds of arrangements of those 4 different things. So here's another picture, of the cis versus trans molecule with that double bond and the arrangement of the groups around them. So with the enantiomers, again, this is when you've got 4 different things attached to a carbon atom and you can have mirror images. I wanted to show you guys, I brought a couple of models here. So you can see the basic geometry, right, of the carbon. Let's say that this gray circle in the middle is the carbon. You've got, this is the tetrahedral shape in 3D. Maybe this might help a bit to see it, right? You got one thing that, if you orient it like this, you've got one group that sticks up and three that kind of stick out and down to the sides. Now, I've got another one here that at first might look pretty similar, right? And I kind of color coded the four different things here. But if you look, right, there's actually no way that I can make these two line up and be exactly the same. So if I line up the green here and then I say line up the blue here, if you look at what happens on this side of the molecule, right, now the pink and the yellow are opposite from each other, right? They don't match up, right? And if I turn, no matter how I turn that, anyway, it's impossible to get all four of the groups to look exactly the same and overlap with each other. So that's what I mean by the enantiomers idea. Amino acids are a molecule that do in fact have four different things bonded to a carbon atom. And in biology, we actually only see the L form of it that's used to make amino acids, which is an interesting fact. All right, next, functional groups. So functional groups are groups of atoms within a molecule that confer specific properties to those molecules. Now, here are a list of the different functional groups that we see in a lot of different biological molecules. This list is actually very important. You should be familiar with it. And you should be familiar with kind of what the structure looks like as well as what its properties are. So, this R here that you see here is kind of the rest of the molecule. So it's sort of like a little tag on a little part of the molecule that, in some molecules we see an OH group, for example. In other molecules, we see what is called a methyl group, a CH3 group. Here are some of the functional groups with carbon double bonds to oxygen. We've got the carbonyl and the carboxyl. The difference being, so in the carbonyl group, this R group here is going to be either an H or another carbon attached to some other things. The carboxyl is where we've got the OH group on it. Amino groups have a nitrogen in them. Phosphate groups have phosphorus in them, and sulfhydryl groups have a sulfur. So again, these have specific properties, like the hydroxyl group is polar, the methyl group is non-polar. Some of them are acidic or basic, okay? And some of them can form hydrogen bonds with other parts of other molecules. So again, we see the importance of these functional groups is that we see those pieces as kind of themes that repeat themselves in various different kinds of molecules. For example, we see some of the functional groups here in DNA, right? And like we mentioned, the hydrogen bonds are actually what holds the two strands of DNA together and makes it a very stable molecule, which is important because these are really the blueprints for the cell. So we want it to be fairly stable.